righty, welcome back everyone. Welcome back. So we're going to be completing chapter 3 with this recording. So, and we're going to start off today's lecture by talking about proteins. So proteins are actually the most diverse macromolecules found in an organism. They are composed of carbon, hydrogen, oxygen, nitrogen, and trace elements that are found on the functional groups, such as sulfur. With proteins, you have the individual monomers, which are called amino acids, which are linked together by the specialized covalent bond known as a peptide bond. So basically through the reaction known as dehydration synthesis, you're taking one amino acid, linking it together, so on, so forth, until you have this very long polypeptide, okay? So basically there are many types of proteins that are found in the biological world, in all the organisms that have multiple functions. We have the examples right here in the table. You have the motor protein, you have defense proteins, you have the metabolic enzymes, you have the proteins involved in gene expression, regulation, which we have an entire unit dedicated to. You have cell signaling proteins and structural proteins, and finally you have transport proteins. So if you take a look at the category and then you see the functions of each, I don't want to go through that. You can go through that on your own. And then there are examples of each of these types of proteins. So if you take a cell, an ordinary cell, and you drain it, about 40% of the dry weight of a cell is protein. And then here's a really important concept again, that structure equals function. So that structure of the protein must remain intact in order for the protein to carry out its function. So we talked about the amino acids being the monomers. What are the amino acids? Basically, you have the amino group, which is the N, 1 hydrogen, 2 hydrogen. You have a central carbon, which we call the alpha carbon. Then you have a hydrogen. Then you have our carboxyl group, which is basically COOH. Okay, so C double bond OH. So you have a carbonyl and a carboxyl, but it's a carboxylic acid group. So that's why it's called the amino acid. Now, the final portion right here is a side chain. Now, here's a tip and a trick I always like to tell my students. These parts will always remain the same. The central carbon, or the alpha carbon, the amino group attached, the carboxyl group attached, and the hydrogen. What distinguishes the one amino acid from another amino acid is the R group, or the side chain. And that is what makes each amino acid different from the other. And there are 20 amino acids. Now, in this class, you're not going to have to memorize them. You will have to memorize them in upper level biochem. So you can see that they are grouped into different properties. So you have the nonpolar, the polar, you have charged and uncharged. And basically, this is what's going to give you the ability of, later on, after you form the polypeptide, the ability of the proteins to start folding. It's the way that the R groups are going to interact with one another when the protein is folded and becomes activated. Okay, so remember the peptide bond. It's a specialized covalent bond. It's formed between the carboxyl group and the amino group of the amino acid. Okay, it is a peptide when it's 50 amino acids or less, and then it becomes a protein when it becomes 50 or more amino acids. For example, when you get to 52 amino acids, insulin is a 52 amino acid protein. What are we doing? Again, you're taking the OH from the carboxyl group, you're taking hydrogen from the amino group, and you're forming this peptide bond right here. And then also you have the formation or the loss of water from these molecules to create it. So remember, dehydration synthesis or a dehydration reaction. Okay, the structure of proteins, there are 4 main structures that you have to just know for this class, okay? The primary structure is basically the linear sequence of amino acids. It is the primary structure. Basically, you have the amino acids, for example, histidine, leucine, tyrosine, proline, arginine, linked together, histidine, leucine, tyrosine, proline, methionine, and then that is basically the simple primary structure. It's just a sequence of amino acids. When you have the hydrogen bonding occur between the R groups of the peptide bonds, of the peptide that you created, then you have specialized folding occurring, and that leads to the secondary structure of amino acids. And you have 2 types. You have an alpha helix, which is right here. Again, you can see the hydrogen bonding, hydrogen bonding, hydrogen bonding. That is leading to this unique helical shape we call an alpha helix. And then here you have a beta plated sheet like an accordion. And again, the reason that you have the formation of a beta plated sheet is, you guessed it, hydrogen bonding. Hydrogen bonding. Okay? Now, this is where it's going to get a little more complex, but if you just pay attention, you're going to be fine. The tertiary structure of amino acid is comprised of alpha helix, beta pleated sheets, and even a sequence of amino acids coming together to form a 3-dimensional shape. And basically what's involved in the formation of a tertiary structure are the following forces. You have van der Waals interactions. That's number 1. 2, you have the hydrogen bonding, which is again, it's the same feature that we had in the secondary structure. You have the disulfide bridges that form. You have hydrophobic interactions. So again, the hydrophobic R groups of the amino acids want to interact with one another. And then finally, you have ionic bonds. Those 5 forces together will create this amazing tertiary structure of a protein. Now, the tertiary structure is the finalized structure of an individual protein. So basically, you can have a 1,200 amino acid protein right here that you can see. Again, here's the alpha helix, beta pleated sheet, and then you have the sequence of amino acids. Okay, amazing. Finally, the final structure, basically what you're doing is you're taking multiple tertiary structure proteins and you're holding them together, okay? This basically will form a highly specialized and highly functional protein. For example, an example of a quaternary structure protein would be hemoglobin, okay? Hemoglobin is a great example. Remember that hemoglobin is the protein that is found in your red blood cells that is responsible for binding to oxygen so that you have the ability to deliver oxygen to the tissues. All right, So here's the primary structure again, sequence of amino acids. So I basically went over it with the overview slide. So it just gives you a little further breakdown if you want. The secondary structure, there you go, at the hydrogen bonds. Again, the formation of an alpha helix or a beta pleated sheet. You have the tertiary structure of a protein. So the different forces that I talked about, the hydrophobic interaction, the hydrogen bonding, disulfide bridges, the ionic bond, and then the van der Waals forces, okay? And then finally, two or more polypeptides, so two or more of the tertiary structure proteins coming together, okay, will form this amazing complex protein. Okay, very, very important. We're gonna talk about proteins again in chapter 5, okay? So the shape of the protein again determines its function. So if you have a disruption in the shape of the protein, that's going to lead to the loss of the function. This is called denaturation, okay? So you take a regular egg, you're gonna make breakfast, okay? You add heat to it, and there you go, you create the sunny side up eggs right there, okay? You've denatured the protein in those eggs, specifically albumin, which is the main protein found in eggs. Now, this actually is a irreversible denaturation, okay? But we do have reversible denaturation. And I really like to talk about this example to students to give, again, to make you understand that denaturation of proteins occurs in humans. And we have it occur several times. So, for example, let's say that you get an infection, and I know that it's hitting close to home right now, talking during the global pandemic of COVID-19. But let's say you get infected with, I'm not going to say COVID-19, I'm going to say streptococcus, okay? Strep pneumoniae, which is the causative agent for strep throat, okay? You get strep throat, you catch it, it's going to lodge itself in the respiratory passages, in the pharynx. And basically, what happens is your body reacts through an evolutionary process to increase its temperature. Basically, the fever response. The fever response is an evolutionary response because all the organisms that cause pathology or disease in humans have an optimal temperature of, you guessed it, body human normal average body temperature, 98.6 degrees or 37 degrees Celsius. So it's 98.6 degrees Fahrenheit or 37 degrees Celsius. Now, your body recognizes that there's this foreign invader. Signals are sent throughout your body, and then basically it sends a signal to your hypothalamus, which is a structure that is found in the brain, which we'll cover when we get when you get to 201 and 202, human anatomy and physiology. The hypothalamus will then start slowly increasing the temperature of the human body so as to destroy the bacteria. Now, when you increase the temperature of the human body, you are going to affect a lot of structures. You are specifically going to affect proteins that are involved in catalyzing your hundreds of trillions of reactions. Okay, so that is what leads to the feeling of headache, malaise, nausea, bone ache, muscle ache, whenever you have the fever, because you have a normal protein, for example, an enzyme in your body that's catalyzing, for example, the breakdown of acetylcholine between your nerves and your muscles, which allow you to move, okay? So you break that down, you denature it, okay, that means the acetylcholinesterase, which is the enzyme that catalyzes the breakdown of acetylcholine, stops working. Therefore, you have that feeling that you get when you have a fever. So the headache and all that stuff that comes along with it. Okay, what we call constitutional symptoms. Now, when you restore the temperature of the body to the average of 37 degrees or Celsius or 98.6 degrees Fahrenheit, you can actually renature the protein and it can go back to its original shape. Therefore, it can resume its function. Okay, pretty fascinating. So next time you get a fever and you think about it, now you have a biological explanation for all those symptoms you're feeling, okay? All right, so if you have any questions about denaturation or about the protein structure and how it relates to function, be sure to send me a message or jump into the Zoom meeting on Tuesday. All right, so this huge statement right here. Enzymes are proteins, okay? So basically we can say that most enzymes are proteins and the enzymes are going to facilitate chemical reactions. We're going to talk about that in chapter 6, but basically enzymes help to catalyze a reaction by lowering the activation energy required that you have to overcome in order to allow the reaction to proceed. We're going to talk about this more, but the part of an enzyme that you can see here is an active site, which is where the substrate, which is the biological term that we use for reactant. So chemists use the word reactants and biologists use the word substrate, or biochemists use the word substrate. So a substrate will bind to an enzyme and it's going to bind to an exact location on the enzyme known as the active site. And basically, it is the perfect fit for the substrate, the active site. So you can imagine it like a lock and key. So we have a theory of the lock and key fit and the induced fit, where basically the substrate will sit in the active site and then it will form around it. Okay, so again, just to review, we're going to be discussing proteins throughout Bio 181, but I just wanted to give you a review. The primary structure of proteins is just the sequence of amino acids. The secondary structure, you have the creation of an alpha helix and a beta pleated sheet due to hydrogen bonding. Third, you have the tertiary structure formed by the interactions with the side chain. So you have, again, the van der Waals forces, you have hydrophobic interactions, you have ionic bonds, you have the hydrogen bond, and then you have disulfide bridges. And then finally, the quaternary structure basically is taking two or more tertiary structures and combining them together to form this ginormous complex protein. Remember that shape determines the function. So that means if you maintain the shape of the protein, then you'll maintain the function of the protein. And if you disrupt the shape of the protein, then you denature that protein, and denaturation means that you have a disruption in the function of the proteins. Finally, the last topic in terms of proteins that I want, the last point I want to make is that the enzymes are responsible for regulating the metabolic reactions. All right, so please review proteins in your textbook and just go over the PowerPoints again. All right, now we're going to talk about the final group of macromolecules, which we will discuss in great detail in our last unit, and these are the nucleic acids. The nucleic acids are comprised of nucleotides. So the monomers are the nucleotides. They are the sugar phosphate backbone. They have the nitrogen bases, and they have, so those are three. You have the sugar, which is going to be either ribose or deoxyribose. You have the phosphate group, multiple phosphate groups, and then you have the nitrogenous bases or nitrogen-containing bases. Nitrogenous bases are split up into two groups, which we're going to talk about, the purines and the pyrimidines. And then we have some specific characteristics of the nucleic acids. If it's a double-stranded nucleic acid, it is DNA. If it's a single-stranded nucleic acid, it's RNA. Again, this is in biological living organisms. All right, there's a concept in nucleic acids that we have to discuss, and that is what's known as the complementary base pairing. Complementary base pairing. Basically, you take a purine and a pyrimidine. So you're going to have adenine bind to thymine and cytosine bind to guanine. Okay, and that's what you're going to be seeing in the rungs of the ladder of the DNA molecule when we talk about DNA. Okay, basically the thymine and adenine have 2 hydrogen bonds, and the cytosine and the guanine have 3 hydrogen bonds. And the importance of this is it helps to maintain the diameter, the width of the DNA molecule. All right, so let's look at the differences between DNA and RNA. So first of all, DNA is a double-stranded molecule. So easy, DNA double-stranded, D double, okay? So DNA is a double-stranded molecule. The sugar found in DNA is deoxyribose. And the reason it's deoxyribose is we have this hydrogen highlighted right here because with ribose, you have a hydroxyl group, which means you have an oxygen here, but you don't have an oxygen, so you're deoxy, so you're without the oxygen. That's what it means, deoxy, without oxygen. So you have a hydrogen here on the 2' carbon. We're going to get into that later on in our discussion toward the end of 181. And finally, the bases that are going to be used are in DNA, thymine, cytosine, adenine, and guanine. Okay, so when we look at the RNA molecule, the RNA is single-stranded generally, and we're going to talk about if you take micro, you're going to see some viruses that exist actually have double-stranded RNA. And you're gonna be like, wait a minute, what? How did that happen? Well, viruses are very unique, and we'll talk more about that in micro. Again, the sugar is ribose. So you can see on the 2' or carbon 2, it's a hydroxyl group compared to hydrogen here. And then the bases, you can see that RNA has uracil instead of thymine. So there is no thymine in RNA. So uracil, cytosine, adenine, and guanine. Okay, so the reason that there's such a short discussion of the nucleic acids is we're going to have an entire unit dedicated to nucleic acid, and that's going to be the final unit for 181. So don't be alarmed if you're thinking, oh my gosh, we haven't even talked about nucleic acid yet. I promised you we're going to have a very long discussion about that. All right, so just for the summary, go through it on your own, the carbohydrates, the lipids, the proteins, and the nucleic acids, and all of their functions in biological organisms, what they're used for, what the different types of biological organisms use, for example, carbohydrates for, what the lipids are used for, what the proteins are used for, and finally, what the nucleic acids are used for. Okay, so that is the end of chapter 3. These are your study guide terms. So again, please put these on note cards, basically to remember and memorize, but then also Remember how everything ties together. Okay, so when you define electronegativity, why is electronegativity important? Well, electronegativity is the reason that you have polar molecules, at least to polarity. See how they're right next to each other? There's a reason they're next to each other. So please, when you're prepping for the exam next week, remember to not just memorize, understand the terms, okay? And then answer these questions. If you can answer these questions, okay, and you can explain it to someone. For example, explain it to your boyfriend, your girlfriend, your brother, your sister, your mom, your dad, your aunt, your uncle, your grandparents, whoever, your roommate that you're stuck with during COVID-19 right now, okay? Tell them, hey, I want to tell you a story. Do you mind listening? All right. Tell them, hey, you want to talk about that bottle of water you're drinking? And tell them how fascinating water is. Hey, you want to talk about that protein you just had in your chicken breast that you had for lunch? Hey, let me tell you why proteins are so important for us. Okay, use it. That's what biology is. It's explanations. Okay, explanations for what you're seeing. Because as biologists, we're always asking the question, how, why does this occur? That's what biology is. You're not just memorizing terms, okay? Please do not just memorize, okay? It's more than that. All right, again, continue with all these questions right here. If you can go through and after you answer these questions one time by looking up the answers through the PowerPoint or through your McGraw-Hill activities or seeing it in the Labster lab simulation or even through a textbook, then go through the questions and answer them without using any reference at the top of your head and see how close you come to the answers that you had written from the material when you had it in front of you. Okay, again, please, please, please continue to prep. Okay, don't just complete the activities to complete the activities. Complete the activities to learn from them. Complete the study guide questions. Again, they're study guides. You're prepping for the exam. All right. Again, continue through. If you're going to, again, remember, there's only 2 chapters right here, these study guide questions. Okay, we still have to cover chapter 4, which is pretty short, and then chapter 5. All right. Okay, so I will see you next time for our discussion on the cell, chapter 4. All right, take care, everybody.